Hello, everyone. My name is Chen. I'm an associate professor in the Department of Biochemistry, Molecular Biology, and Biophysics. Our research mainly focuses on functional proteomic analysis of protein post-translational modifications. In the epigenetic fields, we are particularly interested in discovering and characterize novel protein modifications, histones, and chromatin-related proteins, such as transcription factors and enzymes. We are very interested in understanding how those protein modifications regulate the histone's activities as well as epigenetic programs in the cells. We believe that protein post-translational modifications is a very important means to fine-tune protein functions. And it also serves as a very critical link that a bridge between the cellular metabolic states and the protein functions. So understanding how the protein modifications regulate the protein functions is very critical to discover how the cells can sense the environmental signals and therefore reprogram their epigenetics landscape to respond to those environmental changes. So we currently work on two different areas of protein modification analysis on chromatin proteins. On one hand, we identify and functionally characterize several novel protein modifications on histones and chromatin proteins, such as lysine succinylation and proline hydroxylation. We're working towards understanding how those modifications regulate the activities of those proteins and regulate their epigenetic functions. On the other hand, we're working towards study very complex protein modifications, histones and chromatin proteins, such as ubiquitination. We develop technologies to identify and quantify those modifications at site-specific level to understand how those modifications mediate cellular response to environmental changes such as DNA damage. There are many challenges in studying protein post-translational modifications in epigenetics field. The first challenge that we face is that many of those modifications have very low abundance. Therefore, developing very efficient technologies to enrich and identify those modifications is a very big challenge. The second challenge we face is that many of those protein modifications are highly dynamic and reversible in many cases. Therefore, how to quantify those modifications very reliably at the site-specific level represents a huge challenge. Epigenetic Consortium at the University of Minnesota is a very good platform for researchers like me who are interested in epigenetics and to gather together and share their study results. And also, we can learn the cutting-edge research in the field, such as new technologies and new discoveries in epigenetics. So this is also a great platform for us to share our expertise and also find collaborations in research. My lab has been involved in the Epigenetic Consortium for several years. Both myself and my students have been giving seminars in the Epigenetic Consortium, and also we have participated in the joint grant applications. Recently, we have two papers published in collaboration with Natalia Shredekova's lab to study how the cigarette smoke regulates epigenetics in lung cancer. Epigenetic field is a very interdisciplinary field. No one can be an expert in all the fields. So this consortium serves as an excellent platform for many of us to come together and share our expertise. And very importantly, we can find collaborations so that we can move together with our research projects. So the continuing identification and characterization of protein modifications on histones and chromatin-related proteins will lead to establishment of a network of protein modification events with temporal dynamics on the chromatin proteins. And this network in the future will link to the other information on chromatin, including the chromatin structures and cellular metabolic states. All this information will allow us to gain deeper insights into how the epigenetic programs are managed and maintained during the development of diseases such as cancer and metabolic disease. The Epigenetic Consortium will serve as an excellent platform for us to find collaborations to move forward in this endeavor to identify and integrate knowledge to identify novel functional mechanisms involved in epigenetics regulations of those diseases. At the University of Minnesota, we have a very diverse group of researchers who are interested in epigenetics field. For example, we have people who are interested in the technology development, and we also have people who are interested in molecular basis of epigenetics. and also people who are interested in the application of epigenetics in drug discovery and diseases. So with all those people coming together, the Epigenetic Consortium will serve as a very important platform and catalyst to allow us to have new ideas and move project forwards with new technology development. Anyone who are interested in epigenetics, including the new technology development, as well as finding new collaborations in the field of epigenetic research, I would strongly advise you to come participate in the events of Epigenetic Consortium. It is very helpful for me and my lab to move forward in these directions. I'm sure it will be very helpful for many researchers in many fields that are involved in epigenetics.